Hello, everyone. I am Gustavo Escanzarla. I'm a postdoctoral fellow at the Kelvin Lab, and today we'll be presenting you a short session on how to use the tool Multiple Protein Profiler. This is a tool that can read and profile several proteins at once according to their structural, chemical, and physical properties. This tool was developed in our lab, Dr. David Calvin's lab, which is in the Department of Microbiology and Immunology at Dalhousie University. Our tool, located here in multiple protein profiler, mprotein profiler dot microbiology and immunology.dal.ca. Upon accessing this URL, we find the homepage of our tool. As you can see here, you have two main ways of inputting information to the tool. In that case, proteins that you are inserting in the tool and get their profile. The first model is by entering information here in the text box. Here you can copy and paste all of the proteins that you want and just paste them here. Upon clicking the submit button, you will get all of your proteins profiled for you. In addition, if you don't want to manually copy and paste all of your proteins, you can also select a local file on your computer. Maybe you want to work with the whole proteome of an organism. You can actually upload that into here. Indeed, this is actually what we're going to do here to check and obtain the proteome, all of the proteins encoded by a genome of an organism, and upload these proteins to be profiled by multiple protein profiler. Let's select here in NCBI any organism of our interest. And here I will get an archaean organism. Let's get Halopharax volcani. You can basically enter any organism, and if this organism has sequenced information available, NCBI should be able to fetch these for you. Scrolling down, you will see here that we have the genome of Halopharax volcani. These are archaeon. Upon clicking here on genome, you will be directed to the page with its downloads. Once it's loaded, you'll find information about this particular genome. This is the strain DS2 of Halopharax volcani, and this is actually the strain used as a reference for basically all the genomes of this specific organism. If you click here in download, you will have many options to download the sequences of your organism. Here, we only want to download the reference genome, so we can select in RefSec only. And here, you have all of the genomic information on these organisms. We do not want the genome sequences because we actually want to obtain the proteins that are encoded by these genomes. So we can unselect here genome sequences and only get the proteins. It means that we are obtaining all of the proteins that are encoded in the Halopharax volcani genome. Upon clicking download, this will automatically download these proteins to your computer, and we can extract here and obtain this information. As you see here, NCBI dataset, then you have your data. You can open the GCF, and you will have here your proteins. You can open these with your text edit, and these are all the proteins encoded by the genome of the archaeon Halopharax volcani DS2. If you want to count how many proteins are encoded by this genome, you can see here that you have 3,892 proteins. Now that we have already downloaded these, we can close it and come here to our multiple protein profiler tool. We will choose our file. We'll select our proteins, and we will submit that to the tool for profiling. It will take a few instances as we identify. We have here almost 4,000 individual proteins, but there you go. All of the proteins are here and profiled for us according to different properties: the length, the gravity, aliphatic index, instability index, the stability of the protein, molecular weight, aromaticity, isoelectric point, charge at pH 7. We also have the fraction of secondary structure, molar extinction coefficient, and finally the atomic composition. You have three functionalities here. The first one is to download your profiled proteins as a CSV. So you can obtain here and download that in a CSV. You have additional information such as the entire sequence of the protein. You also have the description, what that protein is supposed to do, and all of the features that we have previously explored. The second functionality is upon clicking in any one of the links that you see here in the header of the table, and that will uh, get you a plot of these particular proteins. We see here that 70% of these proteins are stable, while 30% are unstable. So you can also save these plots as images and use them accordingly. The final functionality here of multiple protein profiler is upon selecting the accession number of individual proteins. So you can choose any one of your interests here, and that will get you a web page with a breakdown information level of that particular protein. You have the protein sequence. You basically have all of the information that was shown to you before in the table, but here you're only dealing with one protein at a time. This is a ring cleaving dioxygenase. You have all of the information about it here. And that's it. This is a short tutorial on how to use the tool, Multiple Protein Profiler, and how to obtain the entirety proteome of any organism of your interest and use that to profile the proteins. Thank you very much. I'll see you in the next time. Peace.